So I thank all of you for coming and a very good morning to all of you. And thank you, sir, for the kind introduction. But before we start, whenever we say genetics, there is a lot of lab and there is clinics. And I'm honored and glad that we are starting together with more lab-based topic. And I will speak about the lab part of the interpretation, and I will have my senior Dr. Kaushik Mandal sir, who will be speaking about the lab clinic, the genotype-phenotype correlation, and take it further as you see in the report. So, just a question, because we have few people over here. So, what do you think happens when you send the DNA from your from your patient to the lab? So, any idea? I know my juniors over there, so I'll pick up my junior over there. So, Prema, can you say what really goes on? Like when the DNA goes to the lab, what is the first thing that we have to do? What happens the first? It's the DNA isolation. You can sit and answer. So basically, we isolate the DNA and we will start a library preparation and we start the wet lab, as we say it. The wet lab is very structured actually right now. There are chipsets, there are kits which are used, and depending on these kits, actually we will get our data. But at the end of the sequencing, this is something we all know, what we will get are raw files. So there's a lot of these ATGCs and a lot of these raw files that are generated. The idea is what happens after these raw files are generated and what happens forward. Because whenever we see these reports, we feel that just our variant has magically come up, but actually what the lab looks. So I will be speaking about data processing variant call. Sir will be taking up variant analysis and the report generation. So it's a huge topic and it cannot be covered in 30 minutes, but it's just a snapshot which I wanted to give. So the DNA sample comes, there's a raw sequence, the raw files are called the FASTA or the FASTQ files, and then they are aligned, and this alignment takes place with the reference sequence, and it looks something like this. So there will be an alignment, and when it comes aligns, there are again tools which are used for these alignments, the SAM tools or the BAM tools, and then after these alignment, we get a file which is called the variant calling format. Now this is the most important file. Whether the lab is doing the right kind of test, everything is in this VCF or variant calling format file. Now, I will be saying in detail in what this file is and how from this file we get the data. But before that, few definitions that need to be covered. One is depth of sequencing. So when we sequence each of these variants, how many times I am getting the variant vertically is my depth, and the particular gene, how well it has been sequenced is my coverage. So the depth and the coverage, these two are the two words which we will be using very often right now, and this becomes very important. So in this case, you see if this is repeated 5 times, so this depth is 5x, while if on the other hand you see it's 2 times, so that is 2x. So can anybody say which one is better? 5x or 2x? 5x. Yes. Why? Because that we are getting, we are more sure. This is massive parallel sequencing. There are number of DNA in the cells, and that particular region is sequenced 5 times, so we are more sure than 2 times. So that is what it is. And if we look at here, see this is my, so these are amplifications happening. So there can be regions which can fail amplification, or there can be a deletion or duplication which might not have amplified. So here you see in this region the amplification has happened, and in that region there is like sort of a block where amplification has not happened. So coverage for that region is low, while coverage for this region is higher. So
we keep it more interactive. Why alignment is required at all?
Yes. Okay.
Why at all alignment is required?
Can we get a mic with us?
And how does the coverage might change? How does the coverage might change? So why alignment is required? Because
to make the sequence a known thing. To make the sequence a known one, because we don't know who it is.
yes, the fragments which have been generated, we do not know from it where it has been generated. So it will be 200 base pair, 250 base pair. So this is short read sequencing. So what we talk about NGS is short read sequencing. So we we read around 200, 250, 300 base pairs, and they are at random. So we need to align it with a reference sequence and try to find out. Then we do something called a pruning. While we generate this BCF file, we do something called a pruning. You know what is pruning, no? Cutting the tree, right? So you have to cut it nicely so that your data is covered and all data is covered nicely, right? So if you do too much of pruning, what will happen? Your coverage will decrease, right? ज़्यादा काट देंगे तो पेड़ छोटा हो जाएगा. But if you don't do correct pruning, then your false read will come up.
So when sir is saying pruning, it's removal of the duplicates. So when we are doing this file, there will be duplicates, like because this is when the SAM tool and PAM tool is converting. What happens is that we get the same thing repeated. So there are tools which will remove these duplicates and give us the exact file. So that is our VCF file. So VCF file looks like this, and it has 3 sections, if I may say. So I think I'm loud enough. So I don't know how much you can see from there. This is a snapshot of a VCF file. So you can see from here to here, there is a double hash thing which is given. So this double hash things are called the metadata lines. So it is actually setting up what kind of file I have and what I want to look into it. Like what were the parameters I used, as sir is saying, for my pruning or whatever. So this is the first line. So you can see here, there is allele depth, there is alternate allele. So there is total depth, number of samples. So these are basically the variables which I am setting. And then we have a header line. So this is the header line that comes with a single hash. So this single hash, you can see there are actually about 11 columns to this header line. And after that is my data labels. So as I said, these are metadata lines. So these metadata lines are what I am setting. So say I will explain it a bit later, but you know what I am setting, like which database I am looking, what kind of depth I want, what is my filter I am using. So these are all mentioned here. So when you get the VCF file from the lab, you know what they are setting and you know exactly whether they are lying or not will be actually clear in this VCF file. So the next part is this header, and you can see there are this is the chromosome number. So this is giving you from where as sir said, we don't know from where it is coming. So the alignment we get from here. This is the exact genomic position, which will change according to the build. So we also have to mention which reference sequence we are aligning. There are 2. So we need to know which reference sequence they are aligning, the build, and this would be the position. And this you can see is the RSID. RSID, as I said, whether this is whether they have an idea that this variant was reported or not. And we normally use a DBSNP, or there are some other databases which are used. So here you can see that if it is reported, they will give this ID that yes, it is reported. So it's pulling it up, and the dot says that it is not reported, or we don't know whether it is there or not. So the next part that comes in is the reference allele. So this is the reference sequence में जो allele है, that's the reference allele, and then comes the alternate allele. I'll mention about what allele is and what it is, but it's just a variation, like whether there is a variation from the original one, that will be my alternate allele. Now you can see that I have the filter which is saying pass or Q10. So what does it mean? So if I set up that my data which I want, I'm setting up the filter at Q10. I'll see what Q10 is. If it passes, if it is more than Q10, then it will come as pass. If it does not pass, it will come as Q10, which means it did not, that particular sample did not pass my quality check. So just a small thing about, So just a small thing about the quality. So what do we mean by quality of this data? It basically means that, so you understand that we are doing a fluorescence signaling, right? So I need to understand whether there is an error in that call. So this is called a Fred score. So what we are looking at is how sure I am for that particular Fred score. When I'm saying it's Q10, it means that my chance of error will be 1 in 10. So when I'm saying it's Q30, which means my accuracy is basically 1 in is about 99.9%, and my error is 1 in 1000. So that is what we call as the Q10, Q30. This is how we set it up. Normally you will see labs will give you a Q30, and that is the filter that we use. So this is now we come up to the header line. So this header line, I explained this as chromosome position sample field, there is also an order, in what order things are there. So this is sample level annotations, and you can see here they have used certain abbreviations like GT, GQ, DP, MP. What does these mean? GT is basically meaning genotype. Then it's giving us the quality of that genotype. Then it's giving us the depth, and then it is giving us whether it's phased or not. So normally in outside we do trio, right? So but here we will do, normally we will do just one like we just do the index case or whatever. So now you see when you look at the samples, what it's written. So 0, and this is, you see, this is a line. We call it a phase. This 0, 0 means it's the reference. So 0 stands for the reference. So 0, 0 means it's reference, and after that you see there is a value. This value is giving me the quality of that particular allele, and then comes my depth, and then comes whether the haplotype thing. So that we leave because it will be a bit difficult for us to understand. Now what do we mean by allele? Just to make things very easy. So we have eye color. So there are different versions of eye color. So these different versions of eye color or hair color are feature of this eye color. So these are different forms or shades. So this is what we call as an allele. So in all of us we have 2 chromosomes, and these 2 chromosomes have the genes, and the genes can have alternate versions of themselves. There's a reference version which is common, and then they can have other forms, and these forms are what we call as the alleles. Now these alleles can be more than one. Like say I have one, say there's an A base, it can be T, it can also be G. So there can be many different forms of that particular allele as well. So how do we understand that is, and also something that we need to understand is heterozygous and homozygous. So what do we mean by that? So this is wild type where there's no mutation and we have the normal one. This is heterozygous where there is one mutation and on the other chromosome there is no mutation. This is homozygous where in the same gradient, in the same allele, in the same position we have the change. So it is homozygous mutant. This can also be homozygous wild type. And then we have compound heterozygous, which can be in the same chromosome, 2 mutations in the same chromosome. That will tell me that it is cis, or it can be in 2 different chromosomes, that will be trans. So if we are looking at it, then we have to understand this, otherwise we will not be able to interpret the data in the right way. So yeah, why? Because if it is compound heterozygous, like if it's 2 mutations in the same chromosome and the other chromosome is normal, then also a healthy protein is being produced. This is especially true for autosomal recessive disorders, because in that case your protein is produced, you will not have a disease. But in this case where there are 2 mutations in 2 chromosomes, there is no protein produced, as a result of which there will be a disease manifestation. Now we come back to our samples, here. So what has happened is, this is the chromosome, this is the position, this is the RSID, this is the reference, this is the alternate allele which we were talking about. This is my quality. They have passed the filter. Now what is this? So there's an info. Now you see, these we discussed. So this is the alternate allele frequency, and this is my depth. Now the format I got is GT, and it says 0. Now see, this is not a linear line. This is a slash. This slash actually means it's an unphased data. We come to what phase data and unphased data is. So and then it says 0, 0, which means that it is the reference allele. Now can we try the next one? If it says 0, 1, what does it mean? Be loud. We are not talking of homozygous heterozygous over here. 0, 1 means on one chromosome we have the reference, and on the other one we have an alternate one. We don't know whether it's homozygous, heterozygous, whatever it is, right? And the last one, what is it saying? 1, 1. The reference allele is not there. There are 2 different ones, right? So now, if we picture it, it's something like this. Now, why this hash and why not the phased line? What is phased and unphased? Do I know from which one this is coming? The one coming from which parent? If I do a trio, when I know that, the way the lab annotates it, it will be this phased line. Okay, so that phased means I have a trio, and now I know from where this 0 is coming and where the other one is coming. So that is when I can say it is compound heterozygous, or whether it's in cis or trans. That is how the lab looks at the data. So again, this is something that we have discussed. So there will be zero. How lab discusses this 1, 0, and there can be many forms of the allele. So the reference allele, which is the most common. Now you will say, what is a reference allele? Reference allele. So it's the most common allele which is in your population. That is why population data becomes important. So this is the reference. Then there is the first alternate allele, second, third, and we scale it as 1, 2, 3. So now when we get it, so this was what I was talking about. So this I discussed it. This is unphased and this is phased. Where I know that one is coming from the mother, one is from the father, we have this phased one, and otherwise we have the slanted lines. Now, how does the VCF file tells us what kind of variation this is coming? So we know that the variations can happen in a lot of cases, and VCF can be of any, like it can be whole genome, whole exome, whatever. Now, we need to also understand what kind of change has occurred. So how does the VCF say? I'll just give examples and then we will just work it out. So if this is a reference and my alignment says T, and here everything is pass and etc. So the VCF file will sort of denote it as C and the alternate being T. So here it's a missense variant that we are looking at, correct? So similarly, an insertion will have, as you can see over here, see the alignment, as Sir said, the alignment is very important, right? In this alignment zone, what is happening, I have at that position, after the C, see there are 3 bases, and here we will take from the reference base, from where it has changed, and we put it as CTAG. So this is an insertion. So wherever the change starts and wherever the change ends, that is the position the first position which you were seeing over here. See, it started from 3. Here also it started from 3. So that is the position from where we will say that the insertion has happened. Now there can be multiple changes. So some samples show certain things, some samples show certain other things. In those cases, we will give you these kind of alternate alleles. The VCF file will come like that. So similarly, there can be large structural variations, and when you see again the positions and the alternate alleles will change like that. Now this is too complicated, right? But we'll not go there. So number of these points, how do we understand what is the allele frequency now? Like alternate allele frequency in your reports in the bottom of the line, you will see there is an alternate allele frequency there. How do they calculate that? So the number of alleles that has come up divided by the total number of the number of alternate alleles divided by the total number of alleles will give you the alternate alleles. Why is this important? Because this is important for you to understand whether this is a polymorphism or whether this is a mutation, whether you should consider this or not. So what we were talking about, these VCF files. So data processing happens, as I said, we are doing the pruning, we are doing the alignment, and then we got the VCF file. But this VCF file when we got, this is not enough. Now from here we have to filter out what we want. That is my data filtering, and that happens through, there are many softwares, but normally the most common used is the GATM. So if we are looking at this, so this is my VCF file, and this file will be uploaded in these softwares where these filtering happens. Now filtering can be hard filtering, or there can be something which we call as the quality score filtering. Mostly lab will do a hard filtering, and they will also do a quality score filtering. Both filterings are normally done. Now what is hard filtering? So we spoke about site annotations, sample annotations. So now I'm hard filtering, I'm setting up my filters for my samples, right? So now so these are my snips, these are for index. There are few basic filters that are used, so we will use that filters. But here comes also the role of using your HPO terms, your filters. So that is very important. So it occurs through Linux system. So you can see when we type this. So this is C by reference. This is my result. And now see, I'm using what filters I want. So here we have to give the command. Once we give the command, we will get a data which is a filtered data. Now when you see a filtered data, this can be sort of uploaded in a Excel sheet. This is what you probably might look at if you ask for the coverage or etc. etc. This is what lab is giving you. Now when you are looking for this, again, this file, how do they do that? So every lab will have their own software for prioritization, how they are doing these HPO prioritizations. Now I'm looking at, say, I have certain HPOs, and on basis of that HPOs, I have prioritized 20 genes. So they are done through a scoring. So once they are done through a scoring, I will get something like this. So this changes for every lab. They have their own pipeline, and once we get it, so you can see here that if this is the case study that had come, we extract the HPO terms. I don't know how much you can see, but we extract the HPO terms from the summary which you give, and on basis of that, the filters have been used. And you see, there are 68 genes.
Did we discuss what are HPO terms?
Yeah.
So, what are HPO terms?
So, what is the important?
এটা আমিও জানি আমার মাথায় এত কম ঢুকছে না
But how many of you have used the PubMed, and have you used mesh terms to search PubMed mesh terms? Right? So they are pre-designed terms. Like you cannot just tell saddle nose or depressed nasal bridge. So there will be so many terms for a single descriptions. So HPO terms are those terms which are specifically for certain descriptions, right? And they might be mentioned in that syndrome or in that disease, in that genetic disorders. Because whenever we do any form of variant classification, we try to find out this gene matches with a phenotype. We have to know the phenotype.
So it's almost like, like you use the ICD terms, like the disease you use that, some almost like that. So we have to use those terms. And when you use those terms, so this is my extracted word. So say this is a gene, and I'm trying.
How will you find out HPO terms? So HPO is human phenotype ontology, right? So it explains what it is. So you can there are certain databases like
This HPO is only there, Phenomizer is there. You can just type and see. Right now there are hundreds of details.
Like we Google synonym of this thing, right? You get the synonym. So you can just put whatever you think like. They will design the HPO terms for you.
Okay, so say when I did the prioritization, see there's a Z score over here. So on basis of that, they have given me this is the gene prioritization. We are not doing a variant prioritization. This is the gene prioritization. So now normally we look at the first 50 genes or the first 25 genes depending on what HPO we are getting. So on basis of that, see if I click over there, then I will get the variant information. So if I am looking at this, now I have the entire variant information. This part Dr. Kaushik Mandal will be dealing, and I leave it to this. But now when I have got these 20 genes or 25 genes, this is where the clinical geneticist will come in and say which one is matching, or you as a clinic looking at the patient will come in and say that yes, this is matching or this is not matching. So sometimes when you don't get the genes, you actually need to look back at what filters they have used and what prioritization they have done, and hence your report sometimes changes on reanalysis. And because if you have new HPO terms, this list will also change. So these are just an example just to show that see this is a graph. This has come as very strong, and when it has come as very strong, you just look at the matches. Even in a very strong one, the phenotype matches are not 100%. So normally we will not get a 100% match, but we have to look whether the criteria which is there are the stronger ones or not, whether we are getting stronger phenotypes or not, and not the lesser ones. So that is how the lab will do genotype phenotype, but it is left on to the clinics to do the rest. Now that one sir will explain. Now one small thing before I just give this over to Dr. Kaushik Mandal sir. It is basically when you are doing these filterings, according to the ACMG, there are certain things that has to be reported. There are 79 genes. If there is a change, it has to be reported. And also there are certain incidental findings. So this is the VOUS, but if there was a pathogenic or likely pathogenic one which is actionable, these incidental findings also have to be reported. So this is the incidental finding. Then we have the secondary findings, which is those 79 genes, and then there are other findings. So just to summarize, how do we go ahead? We align, then we do a discovery, annotation, filtering. This is the filtering for the normal variants, and then we will go for our own HPO terms filtering, and that is how we get the list of the genes. Now I'll request sir to come and deliver the next half of the talk, which is about now how do we understand the variants are important.
can be like is it done manually or
Like I said, these are all softwares. But how you are training the software, say a lab which is very old will have more data to train. A lab which is very new will have less data to train. So it is how you are, this is how you train.
how is AI influencing on this?
So right now AI is actually not involved. Right now it's not involved. It's all programming. It's all
But in
bioinformatics done manually.
why can't AI you have one program with the AI and do everything, and then we will not needing Anil also. Is it a possibility?
Yeah, this is all in some sense it's AI. I mean, in the sense that everything is automated.
So the data pooling is AI.
Format. But this is usually done in different layers. There are different people doing different things, and also rechecking. I mean, there are potentials for errors given that you use it. So like you automate it.
In future, can this all be like AI and rechecking everything when done?
So you need a single program where you just put in your variant and then it is.
But you have to put your
This is
brain
taking
on
like a
it.
step, from the raw data to the identification or a filtering of the variants. So it goes through multiple processes which are iterations basically and
Because
it's
I'm
updation.
hearing it seems that it is an ideal place for the AI to.
No. So the problem is this is case-specific filters you are using. So AI can only do when there is, say, I'm just doing a carrier screening. Many labs have shifted to AI. Say, I'm just doing like, how do I put it? Like, say, I'm just doing a somatic variation. I know this variation. So those AI can function. But mostly you are using case-specific filters. And as Sir said, that I have said this too simply. It's not this simple. The entire process is not this simple.
And then all the laboratories are regulated, so they have to answer to somebody else, and they're the people that are making the rules. They're the ones that are saying you have to do it this way, and everybody. And in the UK, you have to do it a particular way. You can't use AI at the moment.
AI at the moment is not there. Because the bioinformatics, I have oversimplified it. So, if you have suggestions.
मैम some easy bits. So जो आप पहले example दिखाया that 2x step and 5x step on a same person. So these both things are normal or there is some defect in that?
No, it is normal because your
So we run the
entire
same
genome
concept.
will not be covered. So what you see the labs give you is the mean coverage or the mean depth. That's
Suppose
why
gene A is covered 5 times and gene B is covered 100 times. So is there any defect in gene A or that is the normal?
No, it's not gene.
it's not
This
gene.
X is for a variant. One nucleotide, right?
For that variant,
So in a gene,
Yeah.
there will be 2000 variants.
right? Sir?
For one variant, there can be suppose cover is a 5x. So we will not take something less than 30x.
Right, sir.
Right.
Yeah.
So less than 30x is probably not correct.
It's a mistake then.
Mistake of procedure or the variant is defective or that particular variant is absent.
No. So don't think in those terms. So you are overthinking the same thing.
Okay.
So now first you have to see whether your experiment is okay or not.
So procedure error.
No, so yeah, it's like a reading. So when you're reading it, when you're saying that no, it is absent, I mean that region will not be read. So it will appear as a kind of a gap in your.
But not absent, sir. This is something. Five times at least read. So and the counterpart is 50 times read.
Correct. So it is all the chemistries and the regions. So the different things
So
that
you
happen.
have to go
So
back and
you're not
see
getting
the Fred score.
uniform. But on an average in a genome, you get much more actually, the coverage.
Right.
The coverage is much more. Here, just as an example, 2 and 5x has been.
So 5x is something is 5x, then maybe a procedure error or
The more number of times it's read, and you have the same thing pattern appearing, you're more confident that there's no artifact or an artificial this thing that has been introduced. That's why that is the importance of depth. Coverage is different. Coverage is you take
No,
the
not
breadth of the room.
I'm asking for the depth. Sorry, I'm asking for the depth.
Yeah, the depth.
5x
that's
versus
what
50x.
So if your depth is low, it means that variant don't consider.
as a clinician, we just have to know that on average, each region or a variant or a nucleotide has to be read 30 times before we say.
And if less than that, then if less
Yeah,
than
If
that,
it is less than that, then you will have to take other criteria and to know there may be a probability it's a true and the red depth is not covered. So you'll have to take a call on that. But on an average,
So if there is a genotype phenotype strong correlation
yeah.
and you have a lower depth, happens with PKD
So
genes,
it's very
then
important.
you
That's
have
a
to
good
do a
question.
Sanger and see whether you have to revalidate it, whether you are actually looking at
In this whole exam or clinical exam, we also get the chromosome number also, which is
I mean, if your there are उन्हीं बोलने के ना चाचा को
So can we get the chromosome number also on that?
Yes, we got it. I showed that the first hash is the position.
Yeah,
So
right.
the position
So
has the chromosome.
First chromosome number. Just asking. Let's suppose the example is CFHR1, and we know by background that CFHR1 is chromosome 1. So we go back like this and write it chromosome 1, or it is already known from that by itself that it is the defect in chromosome 1.
It's not chromosome 1. So understand we are looking at genetic positions. So we
Right.
saw that there were these positions.
Right, ma'am.
So we are looking at variations in that position.
Right, ma'am.
So here it's not about defect or not defect. When we are looking at VCF5, now what sir will say it's about defect or not defect. First we are filtering. Our first job is to filter. So it might be a region which is not significant only. So it will get out.
Suppose we get a variant on that chromosome CFHR1 chromosome. We also know by on that report only that the defect is in chromosome number 1.
Yes, yes.
Okay. So this is not from the
We
background
know the
knowledge.
position.
But
So everybody, if you keep a note of all your questions at the end, we will make sure we save time.
Yeah, I think that's another way. Like you can all write down the questions.
Keep your questions and we'll make time. I'll cut my talk short.
So these are all Dipanjana slides. If you don't understand anything, it's not my job. So how does exome work? That we get some data, then we have some variant, then we try to fix that variant to the diseases we are thinking, right? So there will be people who are more variant specific, there will be people who are more phenotype specific. Those who are more phenotype specific will try to make that variant towards the phenotype. Those people who are more variant specific, they will tell no, we can't take action on this because it does not look to be that pathogen. So this tussle will always remain, but we have to make and draw a line where, because you know that when we do exome or we sequence large part of the genome, we'll get lot of variations which will be different from the reference sequence, right? That is why we all are different. 1 in 1,000 between all of us will be different, right? So which is actionable and which is not actionable. So there is something to do with the variant. How does the protein changes? Whether the variant will have some abnormalities thereon, right? So family history, genetic testing, laboratory testing, clinical integration, and direct clinical use. So there will be a lot like this, and we might not be able to see. So then came the American College of Medical Genetics. So we use all these things like population database, like a variant which is repeatedly there present in a population is probably a benign one, right? Computational methods, like we will do some computational methods. So probably this is also part of AI also. Use computational methods, so they will try to compute whether this is doing something like a splice site is being generated there, or there is a truncation. Truncation in some genes, the truncation might not be a problem, right? Or there is a folding change in the protein. So these are computational methods. Then there are functional methods, like you do it in the cell line. You do a protein modeling and see how does it. Then allelic data set, like data set which each lab might have, each community might have. Then disease association studies, whether this disease has been already associated with this variant, and variant type, like missense concept, and then segregation. Segregation means whether all the people or all the affected family members have that variation and the unaffected do not have, or the carriers are having that in heterozygous, in autosomal recessive, whether the homozygous or compound heterozygous has both and they have come from one from the father and from the mother in autosomal recessive. So this is what is called segregation data, that will segregate the variant or the mutation or the change in the genetic makeup in the whole family or population, right? And you classify them as pathogenic, likely pathogenic, US, likely benign and benign. So benign and likely benign will be really benign, right? And willingness is the place where we get onto onto our nerves. So we tell all these things. How do we classify them? Then this we have already told. We will not go again. So we'll have some criteria like very strong, then moderate, then supportive or supporting for pathogenic. And for benign again we'll have strong and supportive. So we'll try to put each of these criteria to a variant, right? So because we do not really know if some one variant like for achondroplasia 1138 G2 has been reported in all 99%. So we did not use this criteria, right? So it causes achondroplasia. This variation causes achondroplasia, we all know. But for genes where there are a lot of variations which can cause the same disease, where we need to, and many of them are not reported, we have to put this criteria, right? So what is LOF loss of function? So PVS, and if it is a loss of function, it is very strong. That means there is a truncating variant, a nonsense mutation, protein gets truncated, or a splice site variation which causes a absolute defect. So they will give PVS1 type. Then like PM1, it is a mutational hotspot. PM2 absent from controls, we have control databases. So we'll put all this and then we'll put a chart and find out. Yeah, okay. So we'll put, so this is the older one. So we'll put in the chart, so it did not come. So common variants, we have told that if it is common in population, it is presence of these genome, 80 and 1000 genome. These are all population databases which we will see. So Dipanjana, where is that? 2 of these and 3 of these will make it. So now when we have very strong and moderate and supportive, then we'll have more supportive, some. So if it is one very strong and one strong, it will be pathogenic, likely pathogenic, like that. So you have more stronger, so you put it pathogenic, then so that table is missing over here. So this is how we look at the databases. So you know online Mendelian inheritance of man, here you can see the genes. So CleanVar, AGMD is our mutation database, right? So these are the databases where we can see whether this variant is being reported with the disease, variant in the gene is reported with the disease. Then, so you see these are the 2 alleles colored differently. So heterozygous, you know that one there is the other is normal. Homozygous is both are abnormal. Compound heterozygous when there are 2 variants. So we don't call it actually compound heterozygous. Compound heterozygous means it is in trans because if there are the 2 alleles, so there are 2 different mutations in the same gene. So why is that case is that 2 strong and so you know this that so what is a triplet codon? Why do you call it triplet codon? So 3 nucleotides will code for 1 amino acid, right? So what is the permutation combination? So 4 to the power 3 will be 64. So there will be 64 combination. Taking 1 start codon and 3 stop codon, there will be 60. So when there are 3 nucleotides, 4 nucleotides, and you make a triplet for 1 amino acid, that how many permutation combinations will be there? There will be 64, no? 4 into 4 into 4. 4 to the power 3. Right? But we have 1 start codon, 3 stop codons, 4 goes, so there will be 60. But amino acid has 20. So why do we need that? this is how we survive, right? This is called degeneracy of code. So most of the time the third nucleotide, if it gets changed, they will code for the same amino acid many times, right? So, this is synonymous. So that means that these are alternate alleles. Most of the time it is not, but there are certain diseases where there can be synonymous mutation but a disease. A very good example is progeria. You all have seen Pa, the movie Pa, right? So progeria is where there is a synonymous mutation but it is disease. So there are a lot of other pathogenic risk disease. Then non-synonymous means missense mutation. That means the amino acid has changed. A frame shift means most of the time a deletion or duplication and sequence of amino acids have all changed. Right? The sequence of amino acid when it gets changed is called a frame shift.
Because I am finding it there from there. So most probably they are finding it there.
So these are the various. So then you tell this synonymous, non-synonymous. Then there can be loss of pathogenicity or gain of function pathogenicity. So how does it look like? Then you classify it as pathogenic, likely pathogenic. So whatever we know is that pathogenic and likely pathogenic are actionable. But remember that DBS can also turn to pathogenic or likely pathogenic there because there are certain other criteria. Like you do a functional study, you have seen many with the same variant disease in your cohort. You have done a segregation analysis, right? So it will give them some points which might change a VBS to likely pathogenic if you are very sure of the pathogenicity. And remember that just because we do not have lot of disease databases, in-house disease databases, lot of variation which are not being reported will be interpreted here, but they might be pathogenic. So this is the same one. So these are how protein structure or which can actually tell you functional analysis to protein modeling can tell you. Okay, same protein structure. Tell you that it is going towards pathogenic or towards benign. Then again phenotyping or functional studies, like you do a in vitro assay. In an animal model you can do and see that yes, this causes a disease. So these are strong functional. So again clinical importance and functional effects. Some depends more on the lab, the other depends more on the clinicians. So when we are at a loss and ACMG fails, then AMP criteria has come. They have tried to put more points, more predictions, which is slightly probably will be a new method with time, but I am not sure how much it helps just now. But some journalists do ask whether we have done this or not. So you put more points from -1 to +1, whether along with the ACMG criteria to tell whether there is benign or pathogenic. So again, you have various other things to look at for this, like Clinton's criteria.
So basically what that Clinton meant was there are certain disease-specific genes, say M by H, we are looking at, so they will have different criteria in different populations. So there can be a polymorphic variant in one population, but it might be a mutation in the other population. It's not actually a mutation, but if it is with another mutation, it can become a mutation. So that particular gene which we had looked, remember? So There are certain polymorphisms which are VOUS, but if you give them a criteria in certain populations and if they are in certain domains of certain, along with another pathogenic or likely pathogenic, they together can make a phenotype, and this you have to know certain times. So ClinGen is basically where you can put in those variants and you can reclassify, like you can repoint them, and you can do that. And all these softwares are free. Nothing is like, all these sites are free. They are not paid sites.
Except for like GMDs and all, CNVs will you tell?
So CNV, I think this is something like in nephogenetics, the relevance of this is low. But if we are looking at say this particular report, say remember we were saying which build or which reference sequence to look at. So there at the top you see it's written as array GRCH38. So that is my reference. And then what it's written is 16p30. So 16 is chromosome number 16. P13.3 to P12.1 is basically my cyto position, which means on the chromosome PR at that particular cytoband I am getting, and then there is written the genomic location. So genomic location to what?
It's not 13.1.
Oh, yeah.
This is PR. One is the region, three is the band, and this is the subband. So yeah, one chromosome, one long and one shorter is divided into various bands, 1 to 3 bands, and each band, region, then each region will divide into certain bands, and one band can be divided up to 3 subbands. But it is not that in nephogenetics you do not need CNV. We need CNVs like you have WAGR and all, which are actually CNV associated disorders. This is a microarray report, but we were talking about
CNV stuff. So when you look at CNV, sometimes this is also something you can get in your exome sequencing. How do you get that? When we are looking at exome sequencing, there is no software to actually look at the CNVs. It is basically a lab-trained pipeline. So as Dr. Roth was saying, that when you are aligning these sequence, if I am getting in certain regions an extra amount consistently, or I am getting a deletion, that region is absolutely not covered. Sometimes there can be copy number variations in these regions. There can be small or large depending on it, but we need to have a very high depth to look at it. That's what Dr. Anil sir was saying. So if we don't have a high depth, we will not know whether it is risked or not. So labs which claim that at 80x they are giving you a CNV, that might not be a true CNV. So for a CNV, you at least need your depth to be more than 100x. If you don't get that, the CNV calling might be wrong. Also, this is a trained pipeline, which means the lab person is doing this through eye estimations. Like when we are looking at an IGV, they are aligning, they are looking at that sequence, and it's a lab-based pipeline. There is still no pipeline. Affymetrix probably has a cast which can actually convert these into these kind of CNVs, but it is still not very widely used. So this is lab pipelines.
So why we need to look at CNVs? Till what we are talking about were single SNVs, right? One small nucleotide or 2 nucleotides or 3 nucleotide variations. They are small scale mutations. But there can be certain areas where there will be large deletions and duplications, but there will be no SNV, right? So the single nucleotide variation won't be there. 2-3 nucleotide variation won't be there. But there will be disease because there is a large copy number variation, large deletions or large duplication, including those exons or those part of the gene. ये चलेगा लेकिन? तो microarray report.
Microarray report. So yesterday people were asking about genome browsers, right? Yesterday when we were having a talk amongst ourselves, like what we were looking at was so when you align, so when you that's the position. See there the position is given, and now I'm looking at the number of genes which are covered in that position. So I will get that whether what are my morbid genes. So what do we mean by morbid genes? Like I'm using too many terms. What are morbid genes?
causing genes morbidity.
Which will have a significant effect, right? So if I'm looking, so when we look at these kind of regions, we need to look what are my morbid genes over there. Also by default, what we are looking at is the positive strand. We also have to look at the negative strand, the other strand also, and see which direction. So you can see over here the directions are given. See, so the frame is this side. So when the transcription frame is this side. So you also have to know what you are looking at, and based on that, then they will give you these are the regulatory features, and if you go down, they will also give you the like what is known. Like you can also set filters over here, like whether it's a 1 star, 2 star, 3 star, 4 star, 5 star, in the sense that whether it's reported, whether it is not reported, you are being like you are just looking at everything, or you are just looking at specific disease ones only. So you can actually filter them, and next slide. So see, these are my alignments. So now you see this is, if I am looking at an IGD this way, so these red signifies these regions are deleted. Now see here, this is ClinVar, we know that ClinVar will give us data on the regions which are disease. So they have a disease association. That's a disease association. What do we call it as a database. So if I say all types, they have given everything. The blue ones are not significant, they are normal. So, but now if we filter it up, so next slide here, if we just filter it up, now we see pathogenic, see there is none. So there's no reported pathogenic region in this phase. So this is Decipher, what we are looking at, this is a Decipher genome browser, and this Decipher is something again which is free. You can just put it, if you are looking at your gene position or variant position, if you give it, they will also give what are the upstream variants, what are the downstream variants, and it can help you understand whether this particular variant or this particular CNP, the loss or the gain, like we say this as a P haplo or P triplo score, whether those will be significant or not.
So next, like we get a report, you see a gene, first you see a gene name, in bracket you see plus or minus. You have all seen NGS report, no? So what is that plus or minus?
Plus or minus is not upstream or downstream.
What is plus or minus?
Plus or minus is not upstream or downstream.
No. So you know how we read the gene, like what is the mRNA, right? So what we call a gene is not actually the coding strand, it's the non-coding strand. So forget about that. So you can read the gene from the PR to the QR. The gene can also be read, a different gene can be there in the same region which actually runs from the QR to the PR. So it denotes in which strand it is. You start with ATG, ATG is methionine, it starts coding, so it starts from PM to QR. There can be a different gene in the same region which can be from this arm to, from Q arm to, in the reverse direction, right? So we put minus in there.
So it's the direction of transcription. So 5' to 3', which way is it is the gene is transcribed. So that is where the plus minus comes.
That all the same sequence, which can be both ways?
Yes, same sequence can be, so there can, in one place of DNA, there can be two genes. There can be one gene in the same direction also, a smaller gene in between that.
The frame is different.
Though we know only 1 to 4% are coding, rest of is non-coding. Yeah, incorrect.
Now, if you have any questions, so this is basically a summarize from how we went there. So BCF file, we got the BCF file. Then from there we will do the phenotype, genotype, and the ACMT criteria, the 5 main criteria. You look at the variant itself, whether it's causing a problem or not. You look at whether it's damaging or not, whether it's loss of function, gain of function, what the in silico tools are telling you, what is the frequency in the normal population. So the lower the frequency over there, Why are we looking at normal population? Why are we looking at, तुम्हारे के लिए तो जानते चाहिए. Why are we looking at the normal population?
Novel variants can be.
It's not about novel variants, it's basically we are trying to differentiate between a polymorphism and a mutation. So what will be a mutation? What is a mutation, what is a polymorphism?
Polymorphism is a change in the genes present. Normal one does not cause this.
So the frequency of polymorphism will be higher in the population. So if I look at a variant and I look at the genome AD or whatever the database, the 1000 genomas have it, in a higher frequency, it will mean it's not a mutation, it's a polymorphism. And we say 1%, so it has to be less than 1%. So 0.0001 or 0.001, then it's basically what we can take it as a mutation, correct? So why is it a problem for us in Indian population? Why do we get so many pure USs? Yes, sir. Yes. Why we don't have, we are having, like we will have in future, but still now we don't